Okay, great. So here we are again after 1 week. So we don't have students among the participants, but still we will have a recorder session so that people will be able to see the recording afterwards. So we have with us Glenn, who presented last week. And in addition, we have as a guest Guillaume Stirneman from CNRS France that will give some kind of scientific introduction on basically how to use steered molecular dynamics to model some relevant mechanobiological processes. So the stage is yours.
Yeah, thank you very much, Giovanni, and yeah, thank you, Glenn, for having me join this masterclass and this topic. And I will show you basically how we used steered MD in the last few years to basically study, given examples in the modeling of mechanobiological processes. Of course, this presentation is going to be very biased towards what we did in our group, but there are many groups working on that in the world. So it's much more broad, and I will give you at the end some perspectives on if you want more information, what you can read about to basically have a broader picture of what's happening in the field. So why are we interested in in these forces in biology? Is because they are very present in many different phenomena. For example, muscles is the obvious example where you have force which is exerted at the molecular level. The muscle fibers are made by very long polyproteins, big one which is called titin. And basically, anyway, it applies force, it feels force, and so it's important to understand what's happening at the molecular level. Another example, which is quite interesting to study, but of course quite bad, is what happens when you have infection, for example, urinary infection, and you will see that force plays a big role in this kind of systems, and I will discuss that at the middle of my talk. And then there are some, experimental realization of force at a microscopic level, and they have been used a lot in the last 20 to 25 years. And these are single molecule force spectroscopies. So I guess Glenn mentioned them already, but this is typically applying force to proteins or to RNA or DNA, one molecule at a time. I don't go into the details of how they do that in the experiments, but this is typically using AFM or magnetic optical tweezers, etc. How we do that, the way they work in the experiment, and so the way we can mimic this kind of setup in the simulation are basically, there are two big families of things you can do. So one way to apply force is to do that with a constant force. In the experiment, it's done using a feedback loop on the apparatus. Again, don't go into the details. And this is something we can do as well in the simulation. So we fix one end of the protein and we pull on the other one at a constant force. And typically they do the experiments on polyproteins, so several repeats of the same protein, and it's a way for them to accumulate more data at once. And typically, if you use a constant force, you see a staircase pattern, and each stair here you see in red, each step corresponds to the unfolding of one domain. Okay, don't focus on the rest of the plot here; it doesn't matter. Just what is important is that it's step by step. And the other big mode of force application, which is basically maybe easier to do in terms of the experiments, and we can also replicate that in the simulation, is to basically pull at a constant velocity. So we extend the two ends of the protein at a constant speed, and then we typically record what is a sawtooth pattern, and each peak basically corresponds to the unfolding of one protein domain. And then the curves you see in orange or purple here correspond to the extension of this domain once it's unfolded. And here you have two different colors, two different mechanical resistances, because you have two proteins in the construct. And this is a real experimental trace. So there are many things you can do using simulation tools to have molecular information. So one thing that we did in my group, and it's been then applied to other systems, and you will see afterwards, is basically to characterize the unfolded state ensemble, which looks trivial, but no one had done it before, at least carefully like that. And actually, when you pull proteins under force, you typically generate structures that are very different from the structures you get if you denature the same protein with temperature or with chemicals like urea, for example. So here everything is at scale, and you can compare the size of the folded protein and of that in urea. Each spaghetti here is a different conformation of the protein, but you see it's typically quite compact. But if you put force, you get much more extended structures, and you recover this extension versus force profile, which is the dashed blue curve, and it's typically following a warm-like chain model, which is a very simple and popular model in polymer physics. And basically, within this warm-like chain model, that what we showed is that what gives the elasticity to a protein is basically the dihedral angles. So at least under this range of forces, tens, hundreds of piconewton, what you probe and what you extend is really the dihedral angles, the backbone dihedrals, and you don't extend bonds, you don't extend angles, you're just playing basically with the elastic properties of your dihedrals. And understanding this is very interesting for many applications. We did 2 applications with collaborators in London, the group of Sergi Garcia Mañez in particular, to basically have information about how proteins can recognize other proteins. So here is the example where in the experiments and in the simulation, you can extend one substrate protein, which is the orange noodle, basically. And to this extended protein, we have the binding of a protein chaperone, which is the one which is folded, and which is basically binding to this very specific motif, which is shown in red here. And basically, the more this chaperone protein binds to the substrate, the less refolding you have of the substrate once you quench the force. Because basically the chaperone is blocking basically this sequence, and then the protein cannot refold correctly. And so typically in the experiments, this is what they record, the right-hand side. The percentage of refolding goes non-monotonously with force. And basically, which means that, for example, here, the binding to the substrate is very strong at 150 piconewton, but it gets weaker if you have low forces or high forces. And to understand why this was non-monotonous, we basically wrote a model to basically estimate the cost for these dihedrals, backbone dihedrals, to go from their value at a given force to the value they have in the binding motif within the binding pocket, right? And by estimating this cost using SteeredMD simulations, SteeredMD trajectories, we could basically recover this behavior, especially the fact that at very high forces, all your dihedrals are distorted. So you have to pay a high price. I mean, not too high, but you have to pay a price to basically get the conformation they have in the binding pockets of this chaperone protein, right? So you see here how we can use these simulations to basically understand this kind of peculiar force dependency of the binding affinity of one protein to another. And this is something that we later extended to another protein, which is now a protease. So the protease here is a small Pac-Man on the left-hand side, which is basically cutting a protein which is extended upon force. And we went a bit further by trying to write some numerical model able to predict the force dependency of this cleavage rate. And so to do that, we basically write the potential of mean force at a given force as being the potential of mean force along this dihedral, backbone dihedral angles phi MC. So it's in panel B. This is the potential of mean force in the absence of force. And this we can get with, standard free energy calculations, right? And when you add a force, you have a term which is added, which is minus F, which is a work, basically, minus F times the distance between the extremities of your residue, which is a function of the dihedral angles, right? So the closer these dihedral angles they are to 180 degrees, basically, the more extended your backbone. And if they are smaller, your backbone is basically more compact. And basically what you get is something which is expected. And I think, Glenn, you will discuss that. I mean, you discuss that in your exercises, which is the fact that when you increase force, you basically tend to extend your dihedral angles, and you get to the region where it's called a PP2, which is the top left corner. And you see here that basically you concentrate your population there because your free energy becomes even more favorable in this top left corner. And by doing such a model, we can basically numerically estimate the dependency of the cleavage rate as a function of force again, by looking at how much it would cost to distort each dihedral at a given force to the value it has in the binding pocket. And we get a fantastic agreement with experiments. So the experiment is basically the black dots, and the simulation, the model is basically the green curve here. And honestly, there is no adjustable parameters. So it's quite a very surprising and a very good agreement with the experimental values. And this is what we did in this work. And again, the exercise we plan for the evolution of dihedral of the PMF with force. Another big area of applications of CRDMD is basically to understand the unfolding mechanisms upon force. And there are many works that have been done since basically the first pioneer simulations by Klaus Schulten and people working with him. And something we did a few years ago was for this metalloprotein, azurin here, binding copper, again in collaboration with the group of Sergi Garcia-Mañez in London doing the experiments. And basically they recorded something pretty unusual when they did these constant velocity experiments. They basically saw intermediate peaks, right? So not only the regular sawtooth pattern here, but when you see in the zoom here, you see like small peaks and basically additional steps in the unfolding. And when you basically plot the distribution of experimental steps that you get and you average that over many realization of the experiments, you see here that you get distributions for very short extensions that are quite unexpected, because typically you would see extension corresponding to the full extension of the protein, and this is typically what you see here in the orange distribution. But they also saw intermediates, so the gray and basically the complementary, which is the blue distributions here. So how do you understand that? And here there is a key input from the simulation to see where this comes from. When we do the simulations, same kind of experiment, basically here this is constant force, but this basically gives the same idea, same molecular picture. We do see the two kinds of steps, right? So 6 nanometer, 9 nanometer, and here is at two different traces. The green one, we stopped the simulation because we saw already the extensions, right? So we didn't care of, about what was happening afterwards. So the important thing is that we were able to reproduce exactly the same steps as we were seeing in the experiments. And now with the simulation, we get a molecular picture of what's happening. And for this protein, there is something very interesting, is that the metal ion basically blocks the unfolding on the way. And so you can isolate some intermediates of the unfolding process. And so here you see the two intermediates that we get. And basically these two intermediates, and this is what I show on the next slide, correspond to unfolding either first from the C-terminal side or from the N-terminal side of the protein. And so if you unfold one way, you first generate this 6 nanometer unfolding, and then the rest of the protein would unfold when it detaches from copper, and the complementary of that when you do unfolding from the N-terminal side. And so this picture, which was basically seen from the molecular point of view of the simulations, was basically mostly from the C-terminal in the simulation at least, and then it was confirmed by further experiments. One very easy thing you can do in the experiment is basically to have a non-copper binding protein, so it can still fold, but it doesn't bind copper. And when you do that, you basically completely suppress these intermediates, which basically shows that this is really due to this copper intermediate that can fold. Another thing that you can do connected to that is basically to see how force basically distorts biomolecular structures and assemblies. And here this is tackling a much bigger system, which was, it was, some time ago, a few years ago, and it was a very big system to simulate. And it's basically a system involved in mechanotransduction. So how basically cells can sense forces from their environments. And this is by looking at the E-cadherin, beta-catenin junction. So here red and gray junction. And basically experimentally, they know that when you apply force, you phosphorylate a site which is next to the binding interface of these two proteins. And when you do the simulations, you actually see that indeed, when you apply force, you basically expose locally a site which later can be phosphorylized, and this is very important then for the regulation of this mechanotransduction process. I don't go into more details because this is a very long and complicated story, but again, if this is something we can tackle with simulations and see what's happening locally in the structure when we apply the forces. I now jump on something which is even more, intriguing and complicated, which is basically the catch bond phenomenon, one which is involved in urinary tract infection, which is something very common, very problematic, and which is mostly due to one culprit, and the culprit is strains, a few strains of E. coli, so bacteria, they can basically attach and propagate in your epithelium, on your epithelium, by binding through very long arms. And these long arms are basically typically 1 micrometer, and they are made of many repeats of the same protein, which is female, and then some specific motifs, specific domains at the end of the arm, so it can basically attach and bind to sugars at the surface of your epithelium, right? And they exhibit very peculiar properties, which are basically very important for the bacteria to survive. So when there is no flow, when everything is, like the liquid is immobile, the medium is immobile, basically the bacteria doesn't bind too much to your cells, so it can basically propagate, it can infect you in many places because it can basically swim around and bind and infect you, etc. But if you basically try to eliminate this bacteria, and in that case, in the urinary tract, this is through miction, right? You go to the bathroom and just want to eliminate this bacteria. They basically attach more strongly when you try to eliminate them, right? So it's kind of counterintuitive, because when you think of a bond, you would think that when you apply force, you can, if you pull on something, you detach the two objects. In that case, if you pull on it, you make it stronger, right? So it's very important for the bacteria to survive and to thrive, but it's also very problematic. And so where does it come from? And this is something you can study with flow chambers. So these are things they do in microfluidics, and they can apply various flows in very small chambers. Or you can also pull on the partners with single molecule experiments, like I showed you before. And the state of the art is the following. State of the art picture is the following. This is the end of this pili, the long arm that basically attached to the binding site of your epithelium, which is in gray here. And so you have two states of this end protein, which is made of two subdomains. So one is associated, and this is what you see when you have basically no flow, no forces. And so the two domains you see, pilin and nectin, are pretty close together, and then there is a weak affinity for the sugar on your cell. But when you apply flow and when you basically separate these two subdomains, you have some allosteric transition to a state which is a strong binder. And so the lifetime of the bond is much increased in that case. And then the protein, the bacteria can basically attach to your cells. So this is a consensus picture on which we worked. And in all that, there is no stirred MD per se, except in one specific aspect, which is very interesting, and which is the one that I'm going to show you afterwards. Yeah, so this is a delta G of binding between the two species, and which is the following. Force basically can separate the two proteins, like I showed you, right? So it can basically, it's some allosteric regulator. But what we found is that force was also reinforcing strong binding in each of the states, associated and separated. So let me explain you how this works here. So basically the x-axis is the opening coordinate of the binding site. And so basically when it's closed, it has a stronger affinity than when it's open. And so you have 3 peaks corresponding to closed on the left, semi-open on the middle, and open on the right-hand side. So for the associated state, this one is a weak binder. So basically the open conformation is more stable, right? More favored. But what you see when you apply force, typically 50 piconewton, is that your PMF is shifted to, is shifting to lower values for close conformation, right? So you tend to reinforce stronger binding in this weak binding state of the protein, which means that before the two domains can separate, you reinforce the population, which is already a stronger binder, right? And so it prevents the protein from detaching before you can separate the two domains. So it's like a two-time effect of force in that case. And the first effect is to basically to reinforce this small population, which is a strong binder, before you can go to the super strong binder, which is the S state. And the S1, which is on the right-hand side, you see that you have a similar effect, and that force is basically even more reinforcing the close conformation. So it makes this strong binder even stronger by basically closing even more the act. I mean, having a higher population in this close conformation, which basically reinforces the interaction with the partner. And so maybe I should have started with that, but these PMFs were obtained using first structure generated from replica exchange, Hamiltonian replica exchange simulations, on which we then perform the scan along this opening coordinates of replica exchange umbrella sampling simulations, and we compared that at 0 force and with a constant force which was added and which was 50 piconewton, right? So it's like a combination of steered MD plus N9 sampling here to basically get this kind of behavior. And I think this is something that maybe Glenn points out in his talk, and that is pretty promising in the field, is to combine steered MD with enhanced sampling strategies to basically have a more accurate picture and a more detailed point of view of what's happening at the molecular level for a lot of these phenomena. And this is my last slide, which is very different from the other ones, which is basically how you can use force to basically compute free energy differences, at least on paper, which is using something which is probably one of the most important non-equilibrium thermodynamics result of the 20th century, which was obtained in 1997 by Chris Dzerzhinsky, which is basically telling you that for a system, if you want to know the free energy difference between one state A and another state of the system B, and again, everything I'm going to say is very simplified, but this is the main idea. If you want to know this free energy difference, you can basically do out of equilibrium work calculation of how you go from A to B, right? So it doesn't need to be quasi-equilibrium. You can be really out of equilibrium. You do that tens, hundreds of times, and you do the ensemble average of this quantity on the right-hand side, which is the exponential average of this basically work. And in principle, if you do that, a lot of times, you do the average, you can get the equilibrium free energy difference between the two states at A and B. And of course, this is very nice because computing free energy differences or free energy in principle, I mean, in general, is pretty hard, right? Whereas, doing things out of equilibrium, pulling too fast is something which we can do very easily in the simulations, in the experiments. And so in principle, you would be able to compute free energy differences based on that. And this work basically in the context of steered MD or in the context of single molecule experiments is basically obtained by averaging, integrating your force over displacement for the A to B transition. And this is something that has been done on single molecule experiments, and there are quite a few papers just following basically Dzerzhinsky work, and then it took them a few years to set up all that, etc. But you can see, for example, this one in Nature using optical tweezers. And this is something that was very promising, but in practice, which is very hard not to implement, but to have working, basically. And the problem is that this exponential average is extremely noisy. And so you get in the end results for the free energy difference, which are very sensitive to the number of trajectories you use and to how you perform this average. So there are some tricks on the way to improve that. But this is something which is extremely complicated. I mean, which remains, not very used because of these limitations. And before and this is basically my conclusion, so yeah, I conclude with that. These are some very recent perspectives on the topic. So the last two are basically by myself, and it gives an overview of what we did in our group, but also of what people were doing in the last few years and what they will probably do in the next few years, the hot topics in the field. I should also mention the second one for mechanosensing, which is from Glenn's group, which is a bit earlier than our papers, but basically from last year, also giving you a very nice overview of the challenges of the field. And the first review, which is from the group of Frauke Gratter in Germany, also one of the big groups in steered MD, and in mechanical properties of protein and functions, protein properties of function. And so it can be a great source of ideas and overview of what people are doing and will probably do in the next few years. And I'm going to stop here just to mention some people involved in all this work. Steered MD, I mean, it's very important to have, the experimental connection to, to compare your results with, and it's also often used actually to explain experimental results. So it's benefiting from many people doing the experiments. And here's a group of Garcia Mañez for this, most of this talk here. Emmanuel Farge at Curie, Julio Fernandez, one of the pioneers of single molecule, who was in Colombia and now retired. And so Glenn at NYU for some projects, ongoing projects and all that as well. And yeah, I think that's all from my side. If you have any question, let me know if you want some more details about some aspects. And otherwise, yeah, I'll leave it to you, Glenn, to keep going on your parts. And thank you.
Okay, thanks, Guilherme.
Yeah, thanks.
So now I think then we'll continue with the correction of the exercise that they assigned last week, right?
Mm-hmm. Yeah, it was great to hear from Guillaume about the applications, because I think maybe the examples I have are pretty simple, but we can illustrate some of these points anyway. So let's see, and hopefully it's not too disorganized. Okay, so first let's go back to this masterclass that I set up. And some of these points were just touched on by Guillaume. So first we talked about how to apply a force in PLUMED. You basically take your potential and then you add a force times some collective variable, which we'll show how to do. And then Guillaume talked about steered MD, which involves usually adding a harmonic potential, which with a moving center. So I'll show an example of that, even though it wasn't in the exact exercises. And then I talked about this fist method for which you can then reweight to different forces. Okay, so let me hide my own meeting commands. Okay, great. Okay, so the first system is a 1D potential with two states, and you saw some examples of this for real systems from Guillaume. So the idea is that if you have this double well potential and you apply this minus F times X, here's the potential in the Plume file. So it has two centers at 10 and 20, supposedly. And by the way, I should mention, I noticed it's my own mistake or something. I guess this is because it's a master class. I can mention that here I compute a distance in using the PESMD function of Plume, then I as my quantity that I want to bias to produce a 1D potential, and its distances can't be negative. So my original plan was to have this centered at -5 and +5, but that was a mistake because distances can't do that. So I made it 10 and 20, but another option is to put components after this and then use just the X distance, which I've done maybe in my previous masterclass. Okay, so then what I mentioned is that in the exercises, we'll do metadynamics just to quickly sample both sides of this potential with the bias, and then I use the grid file to compute these free energy surfaces. And the reason it's not exactly centered at 10 and 20, I think, is because of the smoothing that comes from using the metadynamics kernels, basically the sigma values, which I happen to pick kind of large ones. And so I would be curious to try to refine this and see what's exactly contributes to this slight shift, but I didn't have time when I was making the exercises to do that. So it's something interesting to look into. Okay, so right now we're talking about, let's see. That's all fine, but maybe I need to log back in. So, okay, here we go. I had everything nicely open, but I guess I have to do this again. So first of all, I should mention that the examples from our paper on FIST are in Plume Nest, and I started with those for many of these exercises. Okay, but now we're talking about double well. I gave you this plumed file to work with, this PSMD file. Now we have to run the simulations. In my case, I prefer to, this is again for masterclass type purposes, that, oops, this is the fifth one. Okay. Maybe this one. Okay, so the point is that I assigned is to take the restraint command and apply a force by changing this slope parameter. And that applies a constant force in the plus or minus direction. And I tend to write my Plume scripts when I'm varying some parameter with these things that I replace using sed on the command line. Maybe people do fancier things nowadays. So I actually have this run script, which you can take a look at. Hopefully you can read this, which loops over a bunch of forces and replaces the force value in that Plume file with that force, as well as some output file names. It then puts them into these different folders with where it ran with different forces. And then you can see that there's, for example, this grid file from MetaD that I would try to plot. And then in this script, all I do is load those different grid files, reweight by because I did well-tempered MetaD by this prefactor that you probably learned about in the metadynamics masterclass, and then plot these values. And then you can see that the original one has equal weights, and then these other ones get tilted. So there's lots more data in these files, but we don't need to go into that too much. The next example in the masterclass is a two-dimensional potential, which is implemented here that we made up for our previous paper. And the goal was to use FIST to sample from negative to positive forces. Didn't have to do much for this one, actually, just test it out and use this reweighting script to get the following results. So I want to show you what's going on in those files. Okay, so when you actually run this, what you get are a CV file. This is the normal thing, which has what we are biasing, so X and Y, and the energy. And then FIST writes out this F bar, which is the average force, or not the average force, but the force that's applied from the average over the weights of different forces. This is the total energy of the bias, F bar times whatever you're biasing, in this case, Y. So if you check F bar times Y, it should work. And then this is the not to be confused with the forces that we're interested in applying, but this is the Plume usual square of the applied force from the bias. If you look at these other files, and this is why I'm just going to show you these now, and then I won't for the other ones. Might take a minute to load. If these are too big, I will show them in terminal. Okay, maybe I won't show those right now. Hopefully they'll open in a different one. What they have in them is columns like this, which I wrote in this script. So the number of samples, the CV that you're biasing, this average force F bar that we were just talking about. And then for each one, it has, we talked about this in the first lesson when I was discussing with Giovanni, that there was discrete points at which the force is evaluated just for the purpose of numerical integrals. So if I could get one of these to open, there it is now. You will see that there's force 0, force 1, force 2, and let's see, force 0 is this one, minus 15, and then something minus 14.8, minus 14.6. And if you followed this all the way along and going all the way down the column, you'll see that it goes all the way up to 15 because we're going from minus 15 to 15. This helps do the numerical integral, and they're not spaced evenly. And somewhere in the middle, there is force 0. Okay, and then next to it is the computed observable weights. And this is all you need to reweight your data to a particular force. So all we do here is reweight the observable forces, and you start with some column, and then you just have to move over. So it starts from column 4, and then it goes over every 2 in this case. And you compute some observable weights, and then just like in certain other kinds of reweighting, since we're computing histograms is what we're interested in, you just pass the weights to the histogram function. And then when you then plot it, you have to remember these various things about perhaps about transposing and so forth. Okay, as it says here. Okay, so that's what produces this animated GIF that I just showed. And hopefully this example helps you do reweighting from other sections, which is what I suggested. The third example is a helix in lamps, which should hopefully, I think that adding lamps examples to the master classes are kind of new. So maybe this is a good example of that. In this lamps input file, you can set the number of steps to run and the strength of interaction between beads in the helix on the command line. So you can weaken and strengthen this from the command line and run, then you can run various plume files, one of which would be pulling at a certain strength and one of which might be running fist. If I go over to that notebook, yeah, this one, I show this result of how I had to compute a certain quantity to separate left and right-handed helices, which I did here. And then you can, once you can compute that quantity, you can just reweight the results to different forces using the weights from the observable file. And one other thing just to show, because Guillaume just discussed it, and I'd be curious for his take on this if I did it right, is I also took this toy helix model and I used moving restraint to pull it to fully extended and back to compressed and fully extended and back to compressed over and over again, which looks like, let me show that. So this is SMD, like the following. So using moving restraint and these nice features that everyone at PLUMED has implemented, you can start at the initial length and then equilibrate it with a certain, so basically I'm turning on the bias here, and then from here on out, I'm extending and compressing, extending and compressing. And as I mentioned in the tutorial, this SMD, or rather moving restraint, automatically produces the work in the data. And so all that I did here is compute the average work and variance of the work and compute this average work minus beta over 2 times the variance of the work, which is the first and second cumulant from the Jarzynski log of average of e to the beta w that Guillaume talked about. And I was able to produce this potentially the free energy of extending from length 4 to length 12, which kind of makes some sense that there's basically it's most stable by many KT in this in its helical form where it happens to be short, and then it maybe has like a saddle out in some extended state, and then it really doesn't like to be very long, which is similar to the worm-like chain idea that Guillaume talked about, even though this doesn't have the dihedrals. Okay, and then I ran fist on this system and was able to reweight to different lengths. Okay, so I'd be happy to answer more questions about that. And then finally, my last example is about alanine 10 in water. I tried doing it in vacuum, but interestingly, I think that it's much more stable in the compressed state, so it was hard to pull out. But of course, in solvated, it's much more happy extended. And then I tried to implement basically the same idea from Guillaume's previous PNAS paper that he showed, where we just see how what the effect of pulling is on the dihedrals and the end-to-end distance and radius gyration. So here in the alanine example, okay, let's see. I have run fist from a small force to a big force, and I load these observable files and reweight to certain values. One thing, now I just can talk about a couple things for diagnosing fist simulations. So I show this in some of the earlier examples, but it's good to plot the histogram that you sampled without doing any reweighting to make sure that maybe that you're covering more space than you would have without doing any enhanced sampling. And so here we see for alanine 10 that there's basically a compressed state and an extended state that's sampled when you apply forces from -10 to 100 piconewtons. You can also visualize that in terms of end-to-end distance and radius of gyration, which isn't one of the exercises. And then I extract the columns of the Colvar file, which have all the forces here by jumping along through that observable file. And then I'm able to reweight, pick a couple of those forces and reweight the end-to-end distance histogram. So you get kind of what you might expect, which is that for negative forces, it likes to be compressed. For forces close to zero, it has some maybe characteristic average length. And then as you try to pull it out, it probably follows this worm-like chain idea that Guillaume talked about, although this is a bit short for that. And then some other things I didn't talk about looking at in the write-up, but can show now is if you actually look at what the weights are for each force, one thing you can find is that the idea of the method is that the weights of that basically different forces are experienced. The molecule experiences only one force at a time, but somehow it has the average effect of all of these forces. And so at small distances, small forces have big weight, and at big distances, big forces have bigger weight. So you can hopefully see that somewhere within this colored data if you look at it later. And another thing you can look at is what the average over the weights of the e to the beta fq is, and it gives you some curve like this, which shows similar to what I just said. So in the end, the effective force that you feel is when you're short is negative and big and positive when you're more extended. Oh, this is the wrong window. I have another version of this, maybe. Okay, so I plotted one more thing, something we've been using, but it's not in the paper from FIST. So this is the masterclass aspect that I think is interesting. We noticed after we wrote the paper that if you look at this F bar quantity, which is a force that depends on the collective variable you're biasing, that when things are working well, this goes up and down, just like in replica exchange, you might want your the temperature that you're feeling to do a random walk. Here, we want the forces to be sampled uniformly is the idea of the method, and so you should see this going up and down. And then this lastly is showing that you can, here, this is the quantity that we actually bias that we're reweighting, but here we're just taking some other quantity, the compute phi and compute psi from the trajectory, histogramming it with different observable weights, different values, and then you see the same thing, which I showed in the tutorial, and also Guillaume just showed, which is that this upper left basin is populated when you pull with large forces, which corresponds to an extended configuration. Okay, so I think that covers everything that is within this masterclass. And as Guillaume said, we'd be happy to answer more questions about it. You can contact us or, check out the solutions, which maybe I'll post in my GitHub for it later. And then also the Plume Nest for the FIST paper has a lot of these example files already.
Great, thanks, Glenn. I have actually a question about the first example with metadynamics.
Yes.
So you were commenting about the width of the kernels. I'm not sure I understood the comment. So that's weird to me. Actually, you have minima not in the correct places.
Yes, but maybe I just didn't do it very well. So I'd like to, like as I said, I was interested in your opinion on that actually, but I think that it has to do with the fact that I guess I can't say exactly what the issue is, but maybe when you...
But is that the result of some hills, right?
this is the result of, it's actually in the grid file for this one, but it should be the same.
Yeah, I don't know.
So,
I will
we will
try it as
test
well.
it again.
Yeah.
This is what I was going to say. Yeah, I think it's just something that maybe it's good to be cautious about. Let's say if you're not using metadynamics properly, because maybe you could say that I produced this example in faster than you would for a research paper, right? So you have to make sure things are sensible before you publish them if they're really to be published.
Yeah, I will double check it as well, because it's a bit curious that you have actually both minima are not in the right place.
Yes, they're both shifted in a bit, but maybe I have some issue. It could also be thermostating or some other, problem. This actually turns out to be, I think, a fairly large hill height, not hill height, but barrier. So I don't know if how I work things out. And there's also a bit of, I may have not put the units quite the way that I intended to. So we can look into that and update this later.
Yeah, I will have a look at that as well.
Any of the example files later. Mm-hmm.
Okay, great.
Great. And Guillaume, what do you think of my ratchet pulling example? Is that legitimate?
Yeah, no, I mean, sure. I mean, it makes sense.
It makes sense. I don't know if I can pull that back up, but I think that the one thing, this is where I wonder about doing this in practice. No, this one. Which one is it? So I think this is where you would get in trouble for real. And maybe you have some comment about this. So this is a folded helix, right? And so steered MD, this thing, right? So in practice, the reason that you would do the, this is a toy sample example, so it might be correct. But in practice, the reason you would start from folded and unfolded is because you know what the folded state is, and you can kind of sample that well, and then you can pull it out. And so you would never do this, right? This is like the ligand unbinding case where nobody pulls the ligand into the binding site, as far as I know, right?
Yeah, true. Yeah.
And I think so. Yeah. I think that when I actually pull it back to size 3.9, I'm not sure that it has time to fold or not. To the fullest extent that it can, although it probably does for this.
Have you checked like visually what's happening? Or?
Yeah, it definitely. So I looked at the handedness of the helix, and what happens maybe was in that thing that I scrolled by very fast as it goes to like straight and then left or right kind of randomly. So I think it's kind of okay. But I don't think you would ever do this for a real molecular system.
Yeah, no, I mean, as you say, even for a helix, I mean, it's pretty fast, right? So it probably hasn't time to fully equilibrate at each distance, right? So
exactly. And the other thing is then that
refolding in all this steered MD stuff, refolding, I mean, even for simple objects is very hard, right? So unfolding is easy because you destroy, but to reform takes time. Yeah. So probably not the time of your simulation. Yeah.
And but this is just 10 beads or 12 beads. So I think
Yeah.
So the other thing that maybe people would have to read the papers and see what they learned, but this is obviously a very strong pulling.
Yeah.
So you can see it's tracking the moving restraint perfectly.
Yeah.
So that is, I think, what people recommend, right? But then there's some
there are some cases where you have some lag, right? Because the force has to increase before you rupture some interactions, right? Exactly. So typically, if you do that on a real protein system, it's not exactly following the restraints, right?
Exactly. Yep. And so, or you can't get it all the way to the value that you pick, is my experience.
Yeah. But it depends on how strong is your spring, right? So if it's a pretty soft one, you would see big deviations. Yeah.
Mm-hmm. Yep.
And this is typically what you see if you do like moving restraints with springs. Yeah.
Mm-hmm. Cool.
Constant force is a bit safer, right? Because not safer, but it's easier, I think, to think about it, because force is directly the force applied to your bead, right?
Mm-hmm.
Constant velocity is a bit harder to interpret and to work with.
Mhm.
Although it's actually funny because it's easier to do in the experiments, but you know,
Yep.
the setup is easier.
Okay, good. So I can't find my window anymore to stop share, but that's okay.
Should be middle and top.
No, I hid it. I
Oh,
hid the tools, so now it's fully hidden. But
it's gonna be there forever. Okay.
I'm checking if I can stop your sharing, but I don't know how.
I think if you try to share yourself,
Stop sharing, yeah.
Right. Okay, great.
Okay, great. So thanks a
Yeah.
lot, Glenn, and thanks Guillermo as well.
Yeah, you're welcome. Thanks for
Yes, thanks for
the
the
invitation.
setting it up, and we hope people find this useful.
Yeah. Okay. Thank you.
Thank you to you both, and see you around. And Giovanni, see you in